Good evening, everyone, and welcome to this another of our Charnwood Forest Geopark online public talks, bringing you accessible understanding of our rich heritage in Charnwood Forest, which stretches back nearly 600 million years. My name's Jack Matthews. I'm the Geoheritage Officer at Charnwood Forest Geopark, and I'll be your host for this evening. Welcome to everyone as you join us in the room. I can see we've got friends joining us from Heidelberg. I can see we've got people joining from Massachusetts. And we also have some dear friends joining us from the Discovery UNESCO Global Geopark in Newfoundland, in Canada, our sister geopark. And you might have seen a few months ago us playing a video about the wonderful landscapes and geoheritage you can explore at our sister geopark. And for all sorts of reasons, you're going to be hearing why there's such a brilliant connection between our two geoparks. Just before we get going, and while a few more people are joining us in the online presentation room, for those who have been hiding under one of our many rocks in Charnwood Forest, you might have missed that we've recently submitted our application to become a UNESCO Global Geopark. And we have the backing of none other than Sir David Attenborough, who has endorsed our application. So over the next about 18 months, we're going to be evaluated, and we should know by about April or March 2027. If you'd like to get involved in anything we're doing, from being a volunteer to making a donation to the charitable work that we do, or anything else, perhaps you've got an organisation that would like to work with us, you can find out more at charnwoodforest.org. And at that point, I should say a very big thank you to the National Lottery Heritage Fund, who fund a huge amount of our work and enable us to provide events like tonight free of charge. So thank you to all the lottery players in the audience and the National Lottery Heritage Fund. Now, these talks are not academic talks. They are not just to share science with scientists. They are informal and relaxed. And so I'm recommending you get a glass of something nice, whatever you choose to join tonight's talk. My pairing suggestion is a Chilean Merlot, which was the favourite wine of Professor Martin Brasier, a wonderful gentleman who you are going to hear a little bit more about in a moment. Now, before I introduce our fantastic speaker this evening, I just wanted to run through how things will work. We're going to have the presentation, and then we're going to do all the questions at the end. If you have a question, you can ask it via the comments section in either Facebook or YouTube, and I'm going to be putting up some information about other ways you can send me a question if you have one, and then I'll put them all to Duncan at the end of the talk. The best thing to do when you're thinking of your questions is send them to me immediately, because then they're on the list rather than waiting until the end. Welcome, Sonia, from the Bonavista Peninsula of Newfoundland. Wadiat to you. Lovely to have you joining us, and seeing many friends joining us from all around the Charnwood Forest area as well. And at that point, I am going to welcome Professor Duncan McIlroy, who joins us from Memorial University of Newfoundland in Canada, where he's in his office in St. John's, the capital of the province. Duncan is professor at the university in the Department of Earth Sciences and has a rich and vibrant research group that looks at all sorts of questions, but with a particular focus on the pre-Cambrian and the beasties, including the ones we're going to see today. So a big thank you to Duncan. Thank you for joining us. I hear the winds that have just hit the UK, you sent them over from the past few days.
Yeah, they're coming to about 150 kilometers an hour, I think. It took a while.
Well, thankfully they've weakened a little bit, but we are now feeling the Newfoundland winds hit after they've crossed the Atlantic. Well, Duncan is well placed for all sorts of reasons to talk about the wonderful links between Newfoundland and Charnwood Forest, having done his PhD at the University of Oxford, including work on Charnwood Forest and many other Precambrian rocks around the world. Naming species, doing trace fossils and sedimentology, he's the perfect person to take us and delve deeper into that world. And at that point, I'm going to pop Duncan's slides on the screen. And thank you so much, Duncan. Take it away.
Yeah, so full disclosure, Jack was one of my PhD students quite a long time ago. We're now friends and colleagues. And we share in our history Martin Brasier, who was both of our supervisors. And Martin Brasier is obviously who Charnia brasieri is named after. So what I want to do in the little presentation here is just talk you through a little bit of what we do, and it's all going to focus around one type of fossil. It's all going to focus around Charnia, which certainly the people from Charnwood Forest should know a little bit about. But I'm going to give you a little slightly different take on what it might have looked like and what it might have done. So I'm just going to launch straight in here. It's a bit strange talking to an audience that you can't see as well, so forgive me if I stumble a bit. So anyway, so what I'm going to talk about is how we went about reconstructing Charnia brasieri. And I'm actually going to start with Charnia masoni, which it was originally described from Charnwood Forest. So let me just get into this now, if I can. There we go. So for anybody that doesn't know, the period of time in which Charnia lived is called the Proterozoic, and the last bit of the Proterozoic is called the Ediacaran. So before we get to the Ediacaran, all we have is really single-celled things, maybe some algae. And the Ediacaran is when we first have big multicellular things with complex morphologies. And it's quite a long time. It's about 30 million years before we have the first things like trilobites coming into the fossil record that people might know about. So the time interval is about 575 to about 550 million years ago. Seems like a very long time for a good reason, as it very much is. And just to place you, so as Jack said, I did my PhD on Avalonia, which is an ancient small continent which lived on the edge of Africa, which we used to call Gondwana. That's the ancient continent that it belongs to. And this zone of Avalonia comes through Newfoundland, so I'm sat approximately here at the moment, and you guys are sat in Charnwood Forest about there. So Newfoundland is East Avalonia, and the UK, including Charnwood Forest, is West Avalonia. So in the Ediacaran, so about 565-ish million years ago, Avalonia, so England and Wales, Newfoundland, and some other bits of the US and Canada, were sitting at quite high latitudes in the southern hemisphere. So as I'm sure many of you know, the plates have been moving around the planet. Most of our continents are in the northern hemisphere at the moment. But in the Ediacaran, they were way down at the bottom in the southern hemisphere. So this is a diagram for something else, but I just wanted to show you a few places that I will talk about. I'm here in St. John's, so as close to the UK as we could possibly be. Well, we'll talk a little bit about the Mistaken Point Ecological Reserve. That's on the bottom of the Avalon Peninsula. I'm going to talk about the type locality for Charnia brasieri, which is in Upper Island Cove here, on the other side of Conception Bay from St. John's, and some material from Catalina Dome. So that would be the Discovery Geopark, the sister geopark to Charnwood Forest. So there's a bunch of dates in there that I'm not going to worry you too much with, but there's three formations that I'm going to mention at other times. There's the Mistaken Point Formation, the Trapassi Formation, and the Firmuse Formation. And we've got some dates on here. So the bed that the fossils come from, that's the type material of Charnia brasieri, are approximately 551 million years ago. So that's actually quite young for the Ediacaran for such complex fossils. Okay, so I'm going to start in Charnwood Forest. It seemed appropriate since that's where I'm sure quite a lot of you guys are from. With this diagram here, this is from a paper by Trevor Ford in 1958. And Trevor figured for the first time Charnia masoni. He described it in this paper. And so this is Charnia masoni up here. This is figure 1. This is his figure 2. And down at the bottom of here, there's a disc, inkling of a stem. And it seems to be in continuity with the Charnia. In actual fact, these are two different specimens. This is a little bit of gamesmanship in some ways. This is the disc of Charnia discus, which is a completely different, unrelated form to Charnia. And it's interesting because that idea that Charnia had a big holdfast disc is something that you see throughout paleo art. You see it in scientific papers, that this reconstruction of Charnia as having this big long frond, shortish stem, and a big disc on the bottom is something that was based upon a misconception of what exactly Charnia was and the putting together of two unrelated specimens. So whenever we see reconstructions of the Ediacaran, of these very earliest Ediacaran rocks, they're from the deep marine. So these rocks would have been on the order of 2 to 3 kilometers deep. So right down on the abyssal plain, so the deepest water you can imagine. There's no light in there, which in some ways is helpful to us because it means that it's clear that these things were not plants. They looked like leaves, but they were actually not plants at all. What they are is something we'll come on to in a little while. But they have wonderful fractal-like geometry. So you can see that they're leaf-like things made out of kind of twiggy leaf-like things that are also made out of twiggy leaf-like things. So they're called self-similar or pseudofractal. So the first one of these that was recognized from rocks that were before the trilobite, so Precambrian, actually came from Charnwood Forest. So that's this work by Trevor Ford here. And he suggested it might have been an alga and some other things. It was very unclear. Martin Gleichner, who's an Australian paleontologist, he suggested that they might belong to a group called sea pens that are related to the jellyfish. And then in 1989, there was an interesting proposal from a guy called Adolf Seilacker. And he suggested that these things were actually built a little bit like air mattresses or lilos, as you would call them in the UK. That they were kind of puffy things and they had big chambers inside. And he proposed that they belonged to an extinct phylum. So we don't know of any other extinct phyla, but he proposed an extinct phylum called the Vendozoa. Now, most people consider that that Charnia belonged to a group called the Rangiomorphs, and specifically a family within that called the Charnidae. And current consensus, partly come out of the UK from Frankie Dunn in Oxford, is that these things were very simple, if you like, proto-animals. So when we see these reconstructions, because there's a big disc on the bottom of these, and many of the early reconstructions, and pretty much everything since, has had these things growing upright into the water column. Once humans have a visual image of what something is like, it's very difficult to change that impression of what the organism looked like. So our view of the ancient seafloor invariably has lots of very tall things. And the logic for that is based around the presence of this basal holdfast disc, which we don't, in my research group anyway, don't necessarily adhere to. So this is a view of the Mistaken Point Ecological Reserve seafloor. So this would have been at the same age as the deposition of some parts of the succession in the UK, the Charnia. And what you'll notice from this is that this is supposed to be as if you could go down in a submersible to the 565 million year old seafloor, shine a light on the seafloor, and what would you see? So there's bits of marine snow floating past, so bits of plankton, goop basically, that was settling down onto the seafloor. And what you'll notice in our reconstruction here is that almost everything is laying very flat to the seafloor. And I'll explain the reasons for that in a little while. So this is a beautiful reconstruction that Bob Nichols did. Bob's based in Bristol in the UK. And so we imagined what the seafloor might be like. And so all these white bits in here, the bits of microbial mat, and growing in between them are various things. Most of these are what we call rangiomorphs. So they would be relatives of Charnia. And here's the star of the show, lurking in here. This is the Charnia masoni, which is growing, as you can see, along the seafloor rather than falling down onto the seafloor, which is what a lot of people would have considered that to be. But I'm going to explain my logic in a little while. So for anybody that doesn't know, this is Charnia masoni. So this is the holotype. So this is the first described specimen, the most perfect example at that time of a Charnia. And Charnia masoni has these very beautiful, almost S-shaped branches. We call them sigmoids. So they're sort of tangential to the axis here, and then they become tangential to the margin of the frond up here. So this is the actual rock specimen. This is a reconstruction that Bob Nichols did for us. So this is a 3D model that he created, and he's put it here onto a seafloor that he created using, basically painted it. So that was our sort of starting point for doing some of these things. I just want to show you a little bit of how Charnia is made up. It seems to be quite simple in comparison to a lot of the rangiomorphs, in that it has these S-shaped branches and then just kind of has bars going across in more than one direction. Some of these in Charnia seem to form arcs, and the reason for that is that they're at about 90 degrees. So you can see that they're 90 degrees to the branch axis here, and that because it's curving, those 90 degrees angles become progressively steeper. So they make these beautiful arcs in Charnia masoni, which is really nice. It's actually, to my mind anyway, a character of the species. So I'm going to show you a different species of Charnia here. This is a new species that was described just this last year by one of my students, Giovanni Passanetti. And here you can see one of those branches. So this is Charnia winoni. Winon is a Beothic word for the people who live here and who lived here in Newfoundland before the Europeans. We have a word that means feather, ewinon, and so we call this one Charnia winoni. As you'll notice here, the branches are more stubby than they are in Charnia masoni. So they're almost like a trapezium. So that's one branch there. That's the first order branch. And what you can see within that is that there are branches within it. So I've got these greenish colored branches here. So that would be the second order branch. And as you can see, they're made out of branches. So I've got these angled branches here. That's the third order branches. And on some of them, there's a fourth order, which is in this orientation here. And you can see some up here. So you can see that in all cases, they're just like little bars. So they're not like leaves. They're just like individual bars. And I just wanted to show you as well the fidelity of some of the preservation here. This is a fossil from the Matthews surface, which is named after Jack Matthews, who found it. So this is Charnia winoni again. This is the holotype, again, the first described, most perfect example. And that field of view there is 0.7 of a centimeter wide. So these earliest branches that we've got preserved up here are fractions of a millimeter, which is really beautiful. It's also important because these things are preserved. They're impressing onto the seafloor despite being so tiny. And again, we'll come back to that as the story evolves. And then here's the real star of the show. So this is Martin Brasier on the top right. As I said, Martin was my PhD supervisor and Jack's as well. I was actually his first PhD student. I think I was probably a good starter student for him. I was fairly independent. We rubbed along a little bit from now and again, but we remained very good friends up until his death about 10 years ago. Not long before Martin died, he died in a car accident. Not long before he died, he won the Lyell Medal from the JOLSOC of London, which is a very prestigious academic medal. This is him receiving his Lyell Medal here. And one of the things that he got that award for was his contributions to the understanding of this group, the Rangiomorpha, of which Charnia belongs. So people were aware that these things were kind of fractal, but hadn't really sort of put it together into a holistic model. And this is Martin's model. This is not my model at all. This is Martin's model. And so what Martin assumed in his model is that all of the branches in all of the rangiomorphs are biserial. And what that means is that there's two rows of branches. So you can see here that if you took this as a branch, as a first order branch, there's two rows of branches that make that up. And I'm going to take this top example here for you. This is basically something called fractifusus. I'll show you in a minute. It's from Newfoundland. And you can see that of that first order branch, it's made out of second order branches, which are branching off like this And if you zoom in on one of those, you can see that there's alternate branching in there. So a fractal-like geometry for sure. But one of the interesting things about a couple of taxa that Martin was particularly passionate about, Charnia and also a thing called Beothucus, named after the Beothuk of Newfoundland, is that they didn't really just simply conform to this, but they actually showed that those branches could be folded in on one another and also rotated. So in order to explain what you see preserved on the seafloor in Charnia, Martin was basically in this diagram here explaining that we only ever see one side of the frond, so the side that's impressed into the mud, if you like, on the ancient seafloor. And this is how he was explaining how you only get one row of branches impressed into the seafloor. So what Martin is saying is that there are two rows, but they're rotated through 90 degrees, so you're only seeing the edge of one of those. And I'm just going to show you here at the bottom right. This was an experiment that I did to explain rangiomorph branching to my own students. So what I did here was I just took a piece of plasticine and I carved onto it a fairly standard rangiomorph type branch, so your biserial branch. And what I've done with that piece of plasticine then is I've folded it in on itself. So I've furled the edges to make it a little bit like a cigar shape. And so we see these geometries quite commonly in the rangiomorpha, where we don't see those nice curved edges of the branches like this. It's a very straight sort of an edge. And that has an implication that I'm pretty sure that Martin understood, was that if that was convex on one side, it also had a concave side as well. So what I've also done here is I've taken that from C to D here, is I've curved that all the way in to make a little tube. And then in E here, I've rotated that tube through 90 degrees. And what you can see here is that instead of seeing the two rows of branches, you only see one row of branches. So we call this folding in of the side furling, it's Martin's term. And on the right-hand side here, this is furled, but also rotated. So you only see one side of the branches here. So in these models of Martin's, these are conceptual models, that he didn't think that this is what Charnia actually looked like in terms of its proportions or anything. But anything that's fractal is almost impossible to fully draw, because every time you draw anything with a pen and a paper, you've got to worry about perspective. And Martin was a great artist actually in lots of ways, but it is just so difficult. So what Martin's, the impression that you get from Martin's reconstructions here is that, yes, you can see these branches, but once you get to this level here, you don't get the impression that there's actually any volume or space, which I felt as though there must be, either that or that they were just ornamentation on the surface in some way, which didn't seem quite right. So what I decided to do was to follow these rules that Martin had invented, the basis of his Lisle metal, and actually go about trying to do them using digital sculpting. So I had been an administrator at Memorial University for a number of years, and a position that I gave up to come back to St. John's. I was actually running an aquarium because obviously I'm a geologist. But I came back to St. John's, and I found that I had a lot more time than I had for quite a long interval. I worked there for 7 years. And so I decided I was going to do something different with my time, the free time that was not spent answering emails, that I would learn how to do digital sculpting, inspired by Bob Nichols, who'd done some wonderful sculpts for us. So, rangiomorphs. So this is one of the examples of the rangiomorphs. This is the fractifusus I showed you at the top. So this is a spindle-shaped thing. It's pointed at both ends. And this is a fairly typical sort of a rangiomorph branch. So if you can follow my cursor there, this is a single branch. You can see it's made out of branches. So take, for example, this one. And then that's also, in some cases, I can't quite pick one out at the moment, but these are actually made out of biserial branches too. So 2 series of branches at all orders is something that is characteristic of the rangiomorpha. So the reason for that is, I don't think anybody has any great argument with, is that the rangiomorphs were trying to create huge surface areas And they did that by this self-similar fractal-like branching. So this is the lower surface of something that grew along the seafloor. Nobody disputes the mode of life of fractifusus here. So it was growing along the seafloor, and all of these surfaces that you see here, they might appear positive to you, but they're actually negative. So they're scooped down into the sediment a little bit. All of these surfaces were interacting with the seafloor, which is something we'll come back to again in a little while. So how did we go about it? So, as I said, Martin's concept was illustrated as these idealized shapes. So this is a sculpt that I did of Martin's branching. So this is a branch here, and you can see it's got another order of branches imposed here, and I put another order of Charnia branches actually on here. But I wanted to develop this premise that we could understand Charnia better by putting everything into proportion and just following this rule that all of the rangimorphs had 2 rows of branches. They might be filled on one another, but they should always be 2 rows of branches. And so I wanted to know what they would look like. So I started out with Charnia masoni, and by the time I was finished that, I decided to do this new thing, which we ended up calling Charnia brasieri after Martin. And then what I ultimately wanted to do, I had a very able student, Daniel Perez Pinheiro, who was doing some 3D flow modeling. So I wanted to create some geometries that we could put into some of his flow models that I'll show you at the end. So that's where we're going with this. So as I said, I thought we'd start with something that was familiar, and I thought that that Charnia masoni was a great thing to start with because the geometry seemed quite simple. And I was entirely wrong with that, as I am commonly. But this is what I went about doing. So this is the holotype that I've tried to reconstruct here. And so I've taken one of these branches here. This is one of those nice S-shaped branches. And what I've done is I've got it closed on one side, so where the two sides of the branches meet, but open at the other side. And it makes this kind of pea pod kind of a shape with a hole in the middle, which is nice. So the Charnia frond is made out of dozens of those. I haven't counted them. But each of those branches is made out of branches. So there's these purple branches that I've illustrated on here. So I've taken one pair of them. You can see them opposite each other or nearly opposite each other. And so the shape of those is what's describing those first order branches. So they curve up over the surface to make that nice peapody shape. And you can see that they're the nice S shape that's characteristic of Charnia. So one of the things that I want you to notice here is that the axis where these first order branches join is essentially straight. It comes up through here. However, if we track those second order branches, so the branches that make up the first order branches, they curve up and over and down again in that nice S shape. And so what I've done for you here is I've taken away all of the brown ones. I've just left you with a pair of purples. And I've drawn on very, very crudely here the axis. And you can see this interesting curved shape. So this is the same thing in 3 different orientations. And what you can see here is that the next order of branches, you can see that it's got a hole in the middle. So it's following that same rule. It's got an axis, it's got 2 sides to it, and it's describing a space in the middle. So instead of being this rather solid thing that Dolph Seilacher reconstructed with that lilo kind of a morphology, it's something, to my mind, or to following the rules that Martin created, something that's a lot more open. It's got a lot more space inside. And for me, as somebody who's a paleobiologist, this makes a lot more sense to me. I could never really quite get my head around why something that was like a lilo, why would it choose to be like that? Why would it have such a low surface area? Whereas actually, once you start reconstructing it like this, it has huge surface area. So this is my 3D model that I created. So each one of these, every one of these things on here is individually sculpted by me, which took a very, very long time. Had a toddler or infant at the time, and so there was a lot of time sat not doing very much, waiting for him to fall asleep and things with my wife. And so I spent that time actually doing these reconstructions and putting together Charnia as I felt that it would look like in correct proportions, following the rules of Martin Brasier. And what you'll see here is that it's a wonderfully open structure. It's got these kind of tubes that go inside, but if you peer inside here, you can see that there are also spaces inside here, there's spaces down below. This thing has an absolutely vast surface area. So I started doing just the first and the second order branches, and then with some encouragement from a postdoc of mine, Rod, who's a close friend as well, he persuaded me that I should keep going. Instead of just doing the first order branches, I would do the secondaries and the tertiaries. So this is one of those S-shaped branches. So that's the first order branch, the white. This is the second order branch here. So that's the orange here. And I've kept the colors such that it's orange on the outside and white on the inside, just so that you can keep track of where you are. So this is a pair of branches in 3 different orientations. So you can see that nice space in the middle that we call a lumen. You see the spaces in between the branches. And then I drew, or I sculpted the branches over the top of that so that you can see the shapes that they describe. So if you remember, the axis of the first order branches was very straight. The second order branches had that nice curve to them. And then these third order branches have crazy shapes. I think they're really beautiful myself. And so these are branches that have to then not only curve around the shape of the first order branches, but also curve over that complicated surface of the second order branches. So you have these really wonderful geometries. At least I love them anyway. So this was a huge amount of work. I don't remember how many different objects there are in there that I sculpted. It was in the order of thousands for sure. But every single one of those branches is S-shaped. So it tapers at both ends. It forms that nice sigmoid. All of those branches are biserial, so there's 2 sides to every single one of them. And they become progressively more curved the more orders that you describe. And I particularly like this view here. So you can see that those branches are offset from each other like they are in the first order branches in masonry. So they're kind of staggered. And they just have this wonderful sort of croissant-y feel to them that I really like. Somebody wants to make me a croissant like this, I quite like that. But you can see that there's actually a lot of space involved in that. So I took a very deep breath, and I took my 3D model, which had taken me weeks and weeks and weeks to create, and decided to try to slice it up so that I could see what it looked like inside. And this is what it looked like inside. So this is simply following Martin's rules. I'm not making anything up here. I'm just following the rules that were set out for me over and over and over again. So this is a longitudinal slice through one of those branches. So you can see down, this would be the axis of the first order branch here. And you see quite how many surfaces there are within that. This is the transverse surface here. So if you count the number of surfaces, so this is a first order branch, there's actually 16 separate surfaces. There's inside and outside of each of these branches over and over as we go through here. But the interesting thing is that only one of those surfaces is actually attached to the seafloor. That's this lower surface in our models anyway. So this is something very, very different to certainly what Dolf Seilacker envisaged for the Rangiomorpha, Charnia in particular. And it wasn't an easy thing to really get around. And Martin's obviously struggled with that as well from his drawings. And one of the things that we do as scientists is that we often lean pretty heavily actually on paleo-artists. So artists whose careers are built around reconstructing things like dinosaurs and fishes and arthropods usually. And I work with a wonderful paleo-artist, Bob Nichols, who did some of this early work with me. But they could only do what you explained to them. And these geometries were so complicated, I didn't really feel that comfortable trying to explain something like Charnia to somebody like a paleo-artist. So as I said, I set off on this this three-dimensional adventure in fractal space on my own. And once I got into it, once I started understanding how these things worked, it's actually not difficult to do. It's just laborious to create these shapes. And I just realized quite how brilliant what Martin had envisaged really was when you actually put it into 3 dimensions. So the software that I used was a thing called Nomad Sculpt. I just do it on an iPad. It's very easy. But it's a wonderful communication tool. It helps us to be able to communicate these geometries that I would have really struggled to try to explain to somebody, even if I could understand it. Now, we'll never know if Martin, if this is really what Martin envisaged, or whether this is more than Martin envisaged, because of course he's not with us anymore. But I like to think that he would have, if he didn't necessarily agree with it, he would have at least loved it, because it really is very beautiful. So that's my understanding of Charnia masoni, and basically, if Charnia masoni was built like that, other angiomorphs should be as well, particularly this group, the Charnidae. So one of the great privileges that we have as paleontologists is that we can name fossils after people or things or anything really, and we obviously chose to name this form here. Charnia brasieri. And so this is Charnia brasieri, Martin top right there. So this is a Charnia brasieri. So you can see here that they have those quite similar branches to what we've seen before. They've got these bars that go across. They don't look to be biserial because they're rotated. And you can see, if you look carefully, that there are bars going in the opposite direction as well. So it kind of looks like a Charnia. And so where did we start? Where did we first come across this idea that it might be a Charnia? So we uncovered a wonderful new fossil surface, which is on the other side of Conception Bay from St. John's, about an hour's drive from home. We got permission to clear the site. So this is the very first day that we arrived at the site. There's a little bit of an exposure in a stream where there was actually a Charnia gracilis that had been exposed. And so we came with a reporter and a TV crew actually as well. Excuse me. And we started excavating. We started clearing the surface. We didn't get very far that first day. We cleared about, I don't know, maybe 5 by 5 meters. And as we were doing so, somewhere just around here, I think, just to the right of that wet area, this thing was uncovered. And I was asked, what is that? I said, I think that's kind of like a Charnia, but maybe not. But as soon as I saw it, because I had been spending a lot of time concentrating on some of these architectures, I thought, that is new. I've never seen one of those. And it's really beautiful. And I'm going to call it after Martin because I can. So right from the very start, I don't often do this. I call dibs, as you say here. So I said, this one's mine. I'm describing this. Nobody else can work on that, which is not something, as I said, that I often do. But so I've been working on the site. It's actually bigger than it is in this photograph here, but it's all hand tools. So we're digging, removing the soil from above and exposing this surface, and it has a whole plethora of beautiful things that I'm looking forward to describing through the rest of my career. So I was excited to describe something and name it after Martin. It was about 10 years since he died. It seemed like a good amount of time to maybe do something to recognize that. So we kind of did a little bit of a backtrack here, because what I realized as I was reviewing the literature, so when you describe a new thing, you have to compare it to all of the other forms that are known. And I remembered this specimen, which is actually from Bradgate Park in Charnwood Forest. And I remember it because it was the first Ediacaran organism that I ever saw. So when I was a new PhD student, so my early 20s, with Martin, in my first semester, Martin took me out to Charnwood Forest. It was a beautiful late October day with beautiful sunny day, low angle light late in the afternoon. And this thing just popped out all of a sudden. And this is a thing called, it was already known as a well-known specimen. It was called Charnia grandis at the time. And this was actually the first Ediacaran fossil that I found on my first day in the Ediacaran with Martin. So a long time ago now. Martin did some work on this of his own. As I said, Martin was a very good artist. This is one of his drawings at the bottom here. This is MDB. That's Martin's initials, Martin David Brasier, from 2005. So this is a cast of the specimen. This is housed in Oxford, I think, this particular one. And this is Martin's drawing of it. And what you will notice from this is that instead of having those nice S-shaped branches that are characteristic of Charnia masoni, everything seems to come rather abruptly to the middle of the axis here. So they meet at quite a high angle, but you don't get any idea that these things are curving to go parallel to the axis as they do in Charnia masoni here. So instead of coming and curving, they hit the axis rather abruptly. And this was my worry with it. I actually thought it was going to be a new genus to start with. But the more I looked at it, the more I felt that really it ought to be a Charnia, and maybe there was something that I didn't understand. So this is the whole type of Charnia brasieri, as we described it. So you can see those J-shaped or fulcate branches, and they meet along a kind of a zigzag that comes through here. And it bothered me because instead of having the tapering at both ends, it seems like they're fattest just at the axis here. And I could not for the life of me figure out how that might be. And I was sat in my office, as I am now, playing with some pieces of plasticine, trying to create S-shapes and make them join. And this is actually what I came up with. This is obviously a sculpt rather than pieces of plasticine, which are not fit to share with anybody. But what I managed to do here was I managed to take a Charnia-like branch, so I made that nice sigmoid shape, and instead of having them all just in the same plane, so on the seafloor as it were, I curved them up. And in doing so, they knit together, but on the opposite side, up above the seafloor. And the geometries that produces are those nice zigzag geometries with nice fat ends right up close to the axis. So this is what those branches ended up looking like. So I've taken one of these here and I've pulled it out for you. This is the seafloor. So this is the lower surface that you would see. All of the rest of this is conjectural. So it's hypothetical. So this is the axis here. And instead of staying parallel to the seafloor, as we think it does in Charnia masoni, I curved it up away from the seafloor and across the axis. So you can see here, this is the same thing. The axis curves through this side here. So that's the open side. The open side is here on this one as well. So just three different views of the same thing. So this is what an individual branch of Charnia brasieri looks like. You can see as it rotates around, it has a very pleasing kind of way that it, all of the curves join together. And you'll notice here that something that I've done here is that the two sides are not the same as each other. There's one side that's got bars parallel to each other, and the other one's got a much more sigmoidal shape. And the reason I've done that is based upon actually the holotype of Charnia brasieri. So Charnia brasieri has those secondary branches, and they describe an arc that goes from the axis and curves away. If you remember, Charnia masoni had arcs that were going this way. But when we did this topographic map, so this is a map that my student Helena did for me. Colder colors are deeper, brighter colors are higher. So instead of having those nice curved arcs that characterize that side, you can see that on this side, the branches just go perpendicular to the main axis. And we think that's because this is the lower surface, topographically lower in the specimen as well. And the topographically higher specimen, part of the specimen, the fossil specimen, has a different type of branching, which is really interesting because now we know whether we're looking at the top or the bottom of a fossil. So I put all these things together, and so I put together the models, and I put them on into a graphics package. And what I've done here is I've created a seafloor. I put these Charnia brasieri. I've got the curved axis, and I've joined them together with a bit of artistic license using this sort of zigzagged stolon to join them together. But it does beg the question that if Charnias are different on both sides, why are most of them preserved only with the lower surface? It doesn't make a lot of sense, unless you think they're just growing along the seafloor. So a lot of people have them being upright in the water column, but intuitively you would expect that some of them would twist as they fall, and they would fall the wrong way up. But of the hundreds and hundreds of specimens of Charnia, no, certainly hundreds, maybe not hundreds and hundreds, of the hundreds of specimens of Charnia that are known, all of them seem to fall the same way up, which doesn't seem quite right. So we're getting now to some of the rationale for why we think some of the fossils, particularly Charnia and some of its close relatives, grew along the seafloor instead of having that upright habit that all of the tropes and memes of Charnia that you've undoubtedly seen have been putting forward. So why do we think that Charnia grew along the seafloor? So if these things were growing upright in the water column and we wanted to prove that, we would look to find one preserved vertically. And we've never done that. Now, I admit that's a high bar that we've set for ourselves, but that would constitute proof. Because if something was lying down on the seafloor, it's very unlikely to get lifted up and then have sediment accumulate around it and preserved in a different orientation. So that would be the ideal situation. Nobody's ever found a Charnia in that upright position. Not yet, anyway. Most of the reconstructions of Charnia have that big holdfast on the bottom that I mentioned. But actually, if you look at most Charnia specimens, they don't have a holdfast. So there's a couple here, for example, two little ones here. And neither of those has anything like a holdfast. None of the Charnia brasieri have a holdfast. It's a little bit of a myth that they have this disc at the bottom. When we have things that are upright in the water column, oftentimes they swish as they fall onto the seafloor. So you might have seen in sand dunes, for example, grasses that are blown in the wind make those nice arcs. And we've never seen that with the Charnia either. The depositional setting in which we find certainly most of these early Charnias anyway, is on the deep sea floor. And mostly on the deep sea floor, you have currents that are coming down off the shelf, down the slope, and they flatten things into a single direction. It's actually very common in the Ediacaran of Newfoundland to see Charnias pointing in opposite directions to each other. And that is hydrodynamically impossible for them to have been upright, for some of them to have fallen in the same direction as the current, and some of them fall the opposite direction into the current. It's just not hydrodynamically possible. There are some things like the very high surface area of Charnia brasieri. So Charnia brasieri is about 65, 70 centimeters long. It has a huge surface area. It would have created an enormous amount of drag if it was that docile like a kind of a model. And that's not really practical unless you have a big anchor hold fast on the bottom. If there's any current, you would just get ripped up because of the shear stress. Doesn't make a lot of intuitive sense. We've never seen evidence of two Charnias falling on top of each other. You see lots of them in close proximity, but they never fall on top of each other, which seems unlikely. Another thing that we don't see is we don't see sediment trapped between the Charnia and the seafloor. So this is a flow reconstruction here. This is some sort of a Charnia-like thing. If it was living in this upright condition, you would expect for sediment to accumulate in these slower currents in behind the front here. So when it fell, it would fall on top of some of these sediments, and it would ruin the preservation. Charnias are almost invariably beautifully preserved. They don't seem to have this kind of accumulation of sediment between them and the seafloor. So there's a lot of reasons that don't make a lot of sense. So we actually think that rather than being upright in the water column, that these things simply grew along the seafloor. They grew either into or away from the current to exploit the current. They interacted with the sediment only on one surface, that lower surface, which I'll explain in a little minute. So then we're going to circle back to Martin Brasier, because everything circles back to Martin Brasier in the end in this field. As Jack will probably smile now as I say this, but one of the annoying things about Martin was that he was very, very clever, and he wrote everything down. He had several diaries and notebooks that he kept going all the way through his career. And I found anyway, I'd have what I thought was a brilliant new idea that nobody had ever thought of before. You'd go and meet him and say, so I've been thinking about this. And he'd say, oh yeah, you should look in my notebook from 1977, the red one. And so you'd go and look in his notebook, and sure enough, Martin would have already written it down. Might not have actually acted on it, but he would have written it down. And so shortly after we started doing all of this work, Alex Brasier, Martin Brasier's son, who's also a geologist, he shared this with me. This is a depiction of Bradgate Park from the fossil surface there. And he has a note on here of the very large horizontal Charnia. Why is it horizontal? It can't be due to felling by ash or sand, question mark. I don't know what his alternative model was. I do know that he thought that some of these things were living along the seafloor like we do. So Martin, of course, already thought about what we've been thinking about, which is, in some ways, it's reassuring, but also it's sometimes a little bit annoying as well, because we all like to think that we're wonderfully original. So I mentioned before that I wanted to explore like how these things actually interacted with their environment. So this is a Charnia brasieri right here. This is the seafloor. You can just about see the shape in behind of those branches. So this is a virtual current. This is a simulated current, which is going from left to right. And what you can see is that those branches of the Charnia are capturing that current and eddying it within those branches before spitting it out the other end here. And that is ideal for two things. It's ideal for flushing. So if there's any sediment grains that get stuck in there, it helps to flush them out. But it also irrigates the surfaces of the organism with which it presumably fed. So what was it doing? What was it doing stuck to the seafloor in our model? So this is that cross-section through a single Charnia branch that I showed you before. And there's not a whole lot of ways that animal-like creatures could actually feed. They could have polyps, like little jellyfishes upside down, which doesn't seem to make a lot of sense. I don't think anybody really thinks that with the Ediacaran biotas, particularly the Rangimorpha. They could have absorbed the nutrients across their membranes. So people have suggested that some organisms could actually just absorb nutrients from their environment. They could have also had something like collar cells. So these are cells that things like sponges use for feeding. So they have these little flagellums that beat, they capture food in the collar, but they can also generate their own little currents, which, if you think about it, might actually help to irrigate some of these small spaces in here. Might be interesting. And the other option is that they could have got their nutrition from symbionts, from bacteria that they grew in association with their bodies, or some combination of the above. So this is a model that we've had for a little while. This is a depiction of something a bit like a Fractifusus, not very good. But Fractifusus is something that everybody agrees grew along the seafloor. So this is a good starting point. And one of the things that happens if you grow along the seafloor is that you restrict the amount of oxygen to the seafloor. So I'm sure many of you have been to a beach and lifted up something like a log and looked underneath for creatures or whatever and seen that blackness that's underneath. So that's a reducing environment. It's often rich in hydrogen sulfide. You sometimes get that eggy smell that comes off it. And that would have happened to an organism that lived on the seafloor if it couldn't move. And I should have said before, but none of these rangimorphs could move. They don't have muscles or any means of locomoting. So they would have had a buildup of this stuff, hydrogen sulfide and probably methane as well, underneath their bodies. Now, hydrogen sulfide is not good. Hydrogen sulfide for animals is a toxin. It binds with the mitochondria in our cells, and it stops them from making energy. So if your mitochondria in your cells are not making energy, the cells die. So what it occurred to Suzanne Dufour and I was that, well, they must have had some sort of a strategy to not have this sulfide build up underneath their bodies. And the most obvious way to do that is to provide oxygen. And if you provide oxygen to your lower surface, what would happen is that you would increase the populations of a very specific group of microbes called sulfur oxidizing bacteria. And the sulfur oxidizing bacteria take that hydrogen sulfide, they use it actually to generate their own energy, so they're using that to increase their biomass. And that would have happened at the interface between the organism and its environment. So the microbes were detoxifying, but also creating biomass at the interface between the organism and the seafloor. And what we hypothesized was that these things might have acted as ectosymbionts, so the organisms could actually engulf some of that organic matter from those microbes and use it for nutrition. They could have had those symbionts within their body walls potentially as well. That's possible, and also within the sort of jelly-like area, the mesenchyme or mesoglia of the organism as well. So that would solve one problem with that lower surface. That lower surface is actually a problem because no matter how something is growing along the seafloor, hydrogen sulfide is a problem. And to actually harness that and make nutrients out of it is a very smart way to do it. But one of the specific reasons that we thought about this was that things like Fractifusus, but also Charnia, have these fractal lower surfaces that were obviously designed to have as big a contact with the seafloor as possible. So if they were actually trying to just grow there passively, that hydrogen sulfide would be killing all of their cells on very high surface areas. It doesn't make a lot of sense. But if they're actually trying to harness that microbial ecology below their lower surface, it might make a lot of sense. But remember, that's only one of those 16 surfaces that I modeled. So it's not the only strategy that we think they might have used, but it's certainly one of them. So this is kind of how we settled, really. This has come full circle from actually having a fossil in the field, to taking photographs of it, to creating 3D models, to actually putting it back into its environment in a mode of life that we think it employed. So these are the small Charnia brasieri that characterize the Newfoundland sections. They're living in association with the microbes. You can see the microbes going around them, but they've also got these high surface area volumes up above that, with which we think that they might have been exploiting the currents, probably suspension feeding, probably just grabbing material that came by. Whether or not that was passively just through the membranes, or whether it was through something like collar cells, we don't have any real evidence for. But I'm pretty confident in myself, and I would say that not everybody would agree with me, that these things grew along the seafloor rather than actually up into the water column. So back in 2021, there was only one species of Charnia. It was Charnia masoni. Every Charnia that you saw in the world was considered to be Charnia masoni. In 2022, just as we were about to describe it, Wu et al. Described Charnia gracilis. It's exactly the same material we were going to describe as a new species. We described Charnia winoni that I showed you some examples of in 2024. And this year, we've added to that Charnia brasieri. So that has those J-shaped branches and those arcs that go in the opposite direction to a masonite branch. And we also resurrected a thing called Charnia grandis, which has a zigzag central axis, branches that come off at 45 degrees and at 90 degrees to one another. So the first order branches are at 45 degrees, second order branches are at 90 degrees. So everything kind of lines up into this beautiful rhomboid kind of a shape. It's very difficult to understand them in the field. But we think now that we have 1, 2, 3, 4, 5 species of Charnia. So it was obviously a very successful architecture. It persisted for between 575 and 550 million years ago. So 25 million years of success, basically the same design. Charnia masoni encompasses most of that period of time, actually. And it was obviously very successful until it wasn't, because of course we don't have any of these here on the planet today, only the fossil examples. We don't really know what they were. They were superseded by the Cambrian biota, so by things like worms and trilobites. There's something that happens in that interval between 550 million years and 538 million years, the base of the Cambrian, in which these things died out, very sadly. I would love to see one in the modern world just to be able to test my hypotheses. Of course, none of this happens in isolation. I've had a wonderful research group here for the last 15 years or so who've been working on this kind of things. We call ourselves the Mum Paleobiology Group, or as Newfoundlanders would say, crowd rather than group. And none of this research here would have been possible without Martin. So he was obviously my PhD supervisor, but he was also the inspiration for exploring these geometries in 3D. And that's about it. I just wanted to also say a good luck to Charnwood Forest Geopark. I have no doubt that you'll be a geopark in a couple of years' time. Thank you very much, Jack.
Thank you so much, Duncan, for a fabulous talk guiding us through lots of human history of the study of these wonderful fossils and the fantastic fossils themselves. And already people are commenting in the chat and saying thank you and good luck to Charnwood Forest. So thank you to Duncan and to everyone for your good wishes as we go on what is a relatively long journey for our UNESCO status. Of course, if you have any questions, keep them coming in. We have a few more moments for a bit of Q&A at the end. And I've got a few questions already. Now, the good people of Charnwood Forest are wise and prudent people, and none more so than the fine geopark volunteers. And in many ways, some of their questions predicted some of your later slides. We had a fantastic question from Penny Broomhead earlier on. Such a large surface area would allow diffusion, but of what oxygen, nutrients? Only a few slides later for Penny to follow up with, very clever. But I just wondered, in response to Penny's initial question, if you had, you talked about the hydrogen sulfide and drawing down the oxygen to react with that. I should say, probably pairs well with the Chilean Merlot.
The sulfide, yeah.
Any other thoughts about how these Charnias might have been using their surface area?
Yeah, I think oxygen was undoubtedly a big part of it. I mean, they're living on the deep ocean floor. We don't really know what things like thermohaline circulation was doing. So that's how oxygen gets down onto the deep ocean floor at the moment. So basically from the Arctic. But I imagine that they were low oxygen settings. There are some ways to measure that geochemically, but the data is often ambiguous in the Ediacaran. It's not very clear exactly what's happening with oxygen, particularly in these deep ocean basins. But I would be very surprised if there was an abundance of it. So anything that can be used to irrigate surfaces would have been a great benefit to them, and particularly if they could do what I showed you with the slowing down, that aids the diffusion as well, so that it helps with flushing, but also, you know, that oxygen is passing through, you know, very large surface areas, the same as our lungs, I guess, but in a different way. Yeah, I mean, our lungs are also fractal.
Yeah, it's important to remember that architecture is still used today, even if it's in a different style. And we should say thank you to Roger, who's already written in to say a fascinating talk and very well presented. And thank you to Penny. Just a reminder to everyone, as you ask your questions, there's no such thing as a bad question or a stupid question. Indeed, I think Duncan would agree that the questions we get from the public are much better than the ones we get from our paleontological colleagues. And there is no better example than the question we've got from Neil, which I absolutely love. What would the texture of Charnia have been similar to? You know, if we were to poke a Charnia, what would it feel like?
So it depends what it belongs to. So to my mind, these things are so beautifully preserved. We do have some examples which have been transported, and they do seem to be quite solid. There's not very many materials that they could have been made of. Sponges are made out of a compound called spongin, which is kind of a little bit flexible. So if these things had choanocytes, maybe they were related to the sponges, maybe they would have been flexible like a sponge. If you take that architecture, that S-shaped architecture, which is also made out of all those interior struts, it's actually quite robust looking. It looks like scaffolding. So I think it would have probably squished. they're kind of, or if the current's coming towards me, they kind of create these open shapes. So they might have been kept inflated by the current that was coming towards them. But also they wouldn't want it to have been like torn and flipped open. So they must have been somewhat rigid. And I think that architecture helps. That sort of scaffolding would have helped. They could have been like jelly, but I don't know. It seems to me that there is a little bit of evidence for them having stalks that join everything together, which is not really very compatible with them being related to the jellyfish. So I tend to think they would have been kind of spongy. That's my intuitive take on it. It could have been something like keratin, like forms your nails, but I think that's maybe too rigid. Yeah. It's a very, very fun question. I like that a lot. I like to be able to imagine things like that. So actually partly what drives me as a paleontologist. What I always wanted to do right from starting out as a paleontologist was to try to bring things to life. And I feel like in the last sort of 5 or 6 years, I've been able to try to. I've actually developed some of the skills to be able to try to do that. But imagining what they look like and how they might have flapped in the current would be a wonderful thing to do. There's a new exhibit which is actually been created in Abu Dhabi, which I was involved with, which has some animations. 3-dimensional animations of these things actually interacting with the current that I haven't seen videos of yet, but the graphics designers that I was working with were definitely trying to encapsulate some of that gentle movement of things flapping and opening and closing due to the actions of the current. So it's kind of flexible and elastic.
And I think I had this conversation about texture with Martin once, and we kind of imagined it from the culinary perspective, and thought that it was one of those probably squidgy sea creatures that you'd have probably had to hold together as you cooked it with a light tempura batter, lest when you cooked it, it turned into just goo and bits. So yes, we were thinking you'd chop it up and put a tempura and deep fry it.
Tempura would do great. It would fill up all those holes inside, and just get you a little, and then probably fishy, I guess.
Bit of chili and garlic on top. It's a good way to think about fossils.
Some aioli to dip it in.
How would you have cooked them? We've got all sorts of questions coming in, both on the text and via the comments, and all sorts of people thanking you for the talk. None other than Priscilla Grew is thanking you for the talk. Priscilla Grew, ladies and gentlemen, one of the only women in the world to have a mineral named after her. Priscilla Grew, joining us from the United States of America. Let's go to another question. We've got one here. Any ideas of rate of growth? Presumably very slow. Larger specimens may have been decades old. What do you think about that, Duncan?
Yeah, I have thought a little bit about that because, so as Jack will attest to, Jack spent a very long time here in Newfoundland looking for new fossil surfaces. And every time you see one of those layers in the cliff, it's an ancient seafloor. They're stacked up on top of each other, and very few of them actually have fossils on them. I mean, we think that we're very rich in them in Newfoundland. I don't know, Jack could probably say there's about 25 different fossil surfaces, for example, at Mistaken Point or something like that. And each one of those represents a special set of conditions. And one of the special set of conditions is that it needs to have microbial growth on the surface. So there needs to have been a pause in the sedimentation for long enough for the microbes to have grown along the surface, for things to have settled, and then they have to get big. If these things were more like jellyfish relatives, they can grow very fast because actually they're only really 2 layers of cells with goop in the middle. And that goop is mainly just water. So I'm sure you've heard about jellyfish blooms, and the reasons they can bloom so quickly is because they're just those 2 layers of cells, and then they fill up the volume with mesoglia, which is essentially water. So they could have grown quite fast. Some of the specimens particularly the specimens actually in Charnwood Forest are very, very big. Some of the biggest. Some of the biggest Charnias, for example, things like Prima candelabrum really big. Hylecalulus, also actually Aurora lumina that you showed right at the very start. That's pretty big too for one of the earliest jellyfish relatives. So I don't know. I tend to think that there was lots of nutrients around because nothing really exploiting them very much. There's no grazing. There's no phytoplankton that we know of. So lots of nutrients might have encouraged them to grow quite quickly. There's no predation. There's nothing eating these things, which is important as well. So there's no cropping of the edges of them. Nothing's nibbling the polyps off them or anything. So I think tens of years is maybe the upper limit, because I think any longer than that, I think they would start getting buried in the sort of the background sedimentation. But no, it's something we think about a lot, but it's very, very difficult to actually pin it down in terms of a number, particularly because we don't have modern relatives. We don't know very much about them at all, which is kind of what makes them exciting. It's certainly what makes them exciting for me is that, basically we're arguing what a thing is from first principles, which is intellectually very, very stimulating and very difficult. It's very amusing.
Thank you, Duncan. And a question coming in from Ruth, who says she had Martin as a lecturer at Hull University and also knew Trevor when doing her PhD in Leicester. And Ruth asks, is there any evidence for the attachment mechanism to the seafloor? How was Charnia attached? Or, to add to Ruth's question, do you not need an attachment mechanism if you're growing along the seafloor as you propose? Yeah,
I mean, most organisms produce polysaccharides on their surface. Essentially, ocean snot is what I always call it for my students. So most organisms have like a sticky substance that they secrete on their outside surface. So if they were growing along the mud and they're creating polysaccharides, mucus on their lower surface, and it's fractal as well, so it's got a really high surface area, I think that would have created quite a lot of adhesion. We have some evidence that, so from some fossils from the Fractifusus, which is that spindle-shaped thing that I showed during the talk, some of those in Newfoundland arrange themselves at about 20 and 50 degrees to the current. And we think that in doing so, they're trying to exploit as much current as possible, so having more of a surface area exposed directly to the current. But they tend to avoid being at 90 degrees to the current. And I think because then they would have too much drag and then they would get lifted up. But I think that they could tolerate a fair amount of seafloor current before doing that. And so I use Fractifusus because nobody contests that Fractifusus was growing along the seafloor. So I think Charnia and Fractifusus probably were growing along the seafloor, and it was that adhesion between their bodies, the mucus and the mud that they were growing through that provided that stability. And I'm sure some of them got washed away as well.
And we should say, for all the people joining us from Charnwood Forest and Leicestershire, unfortunately, we don't have Fractifusus in our fine rocks, but we've got a few fossils that they don't have in Canada as well. So together, we get the full picture, and good example of why we need to study both places at the same time. And
Charnia is very much more interesting than Fractifusus anyway.
I have a soft spot for Fractifusus. And a hello to Samantha, who's joining us from our sister geopark in Discovery in Newfoundland. A good question here from Sita1267. How are the two branches connected? Are they growing from a stem? Which I think what the questioner is asking here are the two sides. Is there a sneaky stem hiding that we're not seeing?
I think I might have an image. Or maybe I don't. No, I don't. Yes.
Oh, you've just left the screen share there, Duncan. So you'd need to reshare, and I can put them on. While Duncan is doing that, I will remind everyone that if you want to find out about everything that's going on in Charnwood Forest Geopark, as well as future talks like this, because it is our intention through the winter and into the early spring to continue these online talks, we encourage you to sign up to the newsletter, which you can do on our website at charnwoodforest.org. You can go to /newsletter-sign-up, but you can find it at the bottom of every single page on charnwoodforest.org, and you can sign up to the newsletter. We send you a little e-update once a month, and you get everything that we've been working very hard on, and everything that's coming up in the future. There's going to be a big event coming up in the new year around March, and you're going to want to be on the email newsletter so that you can sign up to that very, very quickly. So do sign up. Duncan,
I can't find it. I wanted to show a specimen that I actually took out of the talk. I'm a bit disappointed in myself. Yes, so there is some evidence in Charnia masonite that there was a little stalk that went through the middle, so above the seafloor. So you don't see it very often. Sometimes when it's you got a little bit of preservation a bit higher up, there's a little stalk that goes through the axis. And I think that each of those primary branches, those S-shaped branches, did have an extension of that stalk that went through, and then it curved around. And I think that the primaries were probably joined together as well, so that some of those, if you imagine the axis of the primary as an S shape, the secondaries I think went across and joined onto the adjacent branch. So, but I can't prove that. Yeah.
And a very astute question coming in from AJ. Is there a chance of you being allowed to uncover more of the Charnwood bedding plains that are currently covered, like you were able to, Duncan, in Newfoundland? Could we use the same techniques that you used at your fantastic Inner Meadow site and transport those ideas and methodologies over to Charnwood Forest?
Maybe. So the Inner Meadow is really special because most of our outcrops are coastal outcrops. These rocks invariably silica cemented, so they're very, very hard, like a piece of chert. And so they weather very, very slowly. So it is possible to kind of crack open the surface and have a new surface underneath. And we kind of rely upon volcanic ashes for that. So the volcanic ash that we think smothered lots of these fossil horizons weathers preferentially. So it weathers is much softer, easier to weather than the surface that has the fossils impressed on it. So it is possible to crack along that. Inner Meadow is really special because the glacier is actually scoured right down to the surface that has the ash on and stopped. So that over the last 1,000 years or so, the weather's got underneath the ash, and so the ash has been pulled off underneath the field. So it's possible, I guess, that the same thing has happened in Charnwood Forest. I don't think you should go bashing away at the cliffs, hoping to expose more of the D-bed or anything like that, because you would create more damage than you would uncover for sure. But I think that there has been, you know better than me, Jack. There have been lots of additional little discoveries of tiny little outcrops around Charnwood Forest here.
Absolutely. On the whole, AJ, we would go and look at the rocks that are already naturally outcropping, and other than cleaning a bit of algae and moss off them, we probably wouldn't do much more than that. And actually, the most important thing is to look at those outcrops all different times of the day with different lighting. That's going to reveal many more new fossils than if we start digging into the ground. And as Duncan's already alluded to, the big difference is we have a totally different kind of soil profile that sits on top of the rocks in Charnwood Forest than that they have in Newfoundland, where actually it's in general a relatively thin layer of true soil, and then some kind of gravels, but quite angular gravels on top of the surface. So you can kind of move those off relatively easily. Whereas in the upper parts of Charnwood Forest, we have what's called head, which is kind of peaty soil mixed with angular bits, and it might be very thick. So it's unlikely once you've moved that you're going to have a beautiful surface anyway. So in general, it's much better to spend our time finding the rocky outcrop that no one's seen before than digging next to the ones that we already know about.
And the thing is, you don't want to do any harm. You would hate for anything. I mean, the reason we're doing all those excavations by hand is if there was an excavator went in there to even just clear the top off, it would give me nightmares. The idea that, you know, that something might get dragged along the surface or scraped and destroy something really valuable. We just can't do that. Do no harm, I guess.
Indeed, yes. Geoheritage should be like medicine. Hippocratic oath. First, do no harm. And after that, we have. I'm going to take one more question. Well, two at the same time. Penny asks, why didn't they? You talked about Charnia from about 575 million years ago to 550 million years ago. And Penny asks, why didn't they evolve? They had millions of years to do it. And Leanne asks, did they, by which I think Leanne means the various species of Charnia, did they coexist all at the same time? So let's learn a little bit about evolution at this point of time, and were they coexisting?
So yes, they did coexist. So in Newfoundland, we have all 5 species, and they do all overlap, I think. Yeah, I think all of them overlap. So they were all in existence at the same time. Charnia masoni is the oldest of those, so that would have evolved first, I guess. But the oldest Brasier is 565, so 10 million years after the first Charnia masoni, which Jack described. As for what happened before, there's I mean, the origins of anything is always fascinating. The first macrofossils that we have at Mistaken Point in the Druck Formation are about 1.8 meters long. So the chances of those being the first ever organisms to have become complex and macroscopic is tiny, in my opinion. A lot of people think that the trigger for settling on the seafloor was something to do with oxygen, and maybe due to us coming out of an ice age in the late Ediacaran. It's called the Gasquez glaciation. There was a challenge there. There was a real challenge in terms of living on the seafloor, which I mentioned. It's that hydrogen sulfide. So they had to solve that problem of how to grow on a seafloor that was porous and not die of hydrogen sulfide poisoning. So it might have been that adopting those symbionts was a prerequisite to actually settling on the seafloor. It's called symbiogenesis. We actually wrote a paper on it in 2021. It's possible that these earliest animals required that before they could settle on the seafloor. Maybe they were present in the plankton before that. That's entirely possible too. The chances of something with this sort of goopy, thin sort of integument or tissues settling from the water column and getting preserved, particularly in these settings where there was constant currents, and the currents that actually cause the fossils to be smothered are fast currents as well. So we think, you know, if there were things that were settling out of the plankton, then they would probably have got washed away, and maybe we just didn't see them. So those are both fascinating questions. But the other thing about Charnia, which maybe didn't emphasize, is that for a species to have persisted for 25 million years is very, very unusual in the rock record. The diversity that we have is very, very low. I mean, I think there's maybe 30 genera known from the Ediacaran, from these older rocks, which is not very many genera to be known the worldwide, and very few species as well. Most of those genera only have one or two species. Charnia is really unusual, and it has those 5. So ecology and evolution were working very differently back then. And I don't have any good answers. I can only highlight what's different from the rest of the rock record. But again, that's kind of what's exciting about the Ediacaran, is that it's not like the modern, it's not even like the Paleozoic, not even like the time of the trilobonites. The ecology was so different. The world was changing so rapidly. There's like changing in nutrient cycles. We'd just come out of a global glaciation where some people even think the oceans froze over. Things that we can't even imagine in the modern world. And in that setting, we have the very first complex macroorganisms, which is just fascinating.
Well, there you have it, ladies and gentlemen. I think that's the absolute best point to end on. What a weird and wonderful world the Precambrian was, and we dipped our toe into it this evening. Thank you to everyone who's joined us live this evening, and to all of you who are watching again as a recording. And thank you for your kind words, both about Duncan's talk tonight and also our UNESCO application. All I need to add is that, of course, if you want to keep in touch, do sign up to our newsletter on our website, charnwoodforest.org. And you can also sign up to be one of our fantastic volunteers, where you'll get the opportunity to see some of these sites and help care for some of England's very oldest heritage from nearly 600 million years ago. Thanks once more to Duncan. It's been an absolute pleasure to host you. Duncan has been on our expert advisory panel for a couple of years now, so it was great to hear your perspectives on our rocks, and I'm sure we'll be hearing from both you and your research team in the future. But at that point, thank you everyone once more, and we'll see you again in the new year for the next one of these talks. Have a very Merry Christmas, and we'll see you soon.